You and good afternoon, everyone. Jumping across. So the company itself is actually pronounced Recce. It does look a lot like Recce. And just as a brief explanation to that, because I often get the pun, hey, connected to the plumbing company? No, we are not.
My brother is a plumber, so my apologies. I looked at it and just went, Reece Plumbing.
Don't worry, it happens all the time. But the theory behind a good reconnaissance, which comes from the army terminology, is to get behind the enemy lines, attack the bad enemy, and get out without being noticed. In pharmaceutical drugs, which is our space, it's get into the human body, eliminate that enemy, in our case, antibiotic-resistant pathogens or terrible viral pathogens themselves, and get out without undue side effects. And that's really what Recce is about. So as an idea of background, I am the CEO. I was the founding investor in Recce, and I've invested in almost every capital round to date, including putting in about another $50,000 on market last week. I was executive director of the board over the last 5 years, and over that time, as the company has evolved, I've had the pleasure of working with Dr. John Prendergast, who's New York Stock Exchange biotech chairman and lead director of a Nasdaq biotech company, with his doctorate in the study of bacteria and blood diseases. So we were thrilled for him to approach us. My co-supporter is Michelle Delizia. She is our Chief Scientific Officer, and she was really there at that stage of that breakthrough of identifying that compound, and we've since advanced it through the typical drug development pathway. So as a company, look, we are an infectious disease business in the midst of a global infectious disease crisis. Our company is focused on bacteria, particularly the hypermutated bacteria, also known as antibiotic resistance or superbugs, as well as emerging viral pathogens, COVID. For our primary candidate, which is an intravascular compound administered to tackle septicemia or blood poisoning, we have a qualified infectious disease designation awarded by the US FDA. That designation, we are the only company in the world that has that, and that actually provides us an additional 10 years of market exclusivity above and beyond our patent position, government-backed. I mean, 48% of the antibiotic market there, 3% of the market here. Who doesn't want government-backed protection? Our other key candidates is focused on burn ulcers, or the infectious ulcer itself once it's burst. That program is a clinical stage compound on patients at this moment, led by world-leading burns expert Professor Fiona Wood, who was actually former Australian of the Year for her burns work. We have a priority 1 test candidate selected by the Australian government. We're working with Murdoch Children's Research Institute against Helicobacter pylori, which is the bacteria that causes stomach ulcers in humans, and a number of other preclinical candidates. So with a strong intellectual property position, as this synthetic technology is really a land grab in infectious diseases, our own manufacturing capability, producing to GMP standard, we're in a position to really be part of that global infectious disease crisis. So as a snapshot, we have 2 clinical candidates. Our 2 clinical candidates is our compound intravenously administered. Intravenously administered because sepsis is bacteria in the blood. Any bacteria in the blood is bad bacteria, and that's where we target it. As you see there, we're just starting our phase 1 human clinical study, which is in Adelaide CMAX facility, and that will run for approximately 12 months, with interim data readouts expected in the 4th quarter of this year. Our second clinical program, as I mentioned, is that topical wound spray, so utilising that same compound but through a spray solution to the infectious burn wound, regardless of where it is on the body. And that program is active at this moment, led by Professor Fiona Wood. We would expect interim data available next quarter, so Q3, with full data available thereafter. We have, obviously, part of our portfolio, a number of other programs. So as an oral solution to get into the gastrointestinal tract, where Helicobacter pylori resides in the upper duodenum, and obviously other preclinical programs. So if we think of the company as an infectious disease company, I think of it much like an umbrella. That umbrella underneath, we have an antibiotic. Well, there's actually no bio in our biotics. We just call it that for simplicity in communication. And the other umbrella, of course, underneath is the antiviral pathogens. Why are we different? Well, all existing antibiotics to date are naturally derived. They're bacterial fungi cultivated out, put against the offending bacterial fungi, and they're really only as good as what you find in nature. We've began with first principles in mind. We don't want to rely on those natural processes. If you think from a mechanism of action perspective where you have the bacteria and you have the antibiotic, that mechanism of action, much like a lock and a key coming together, may work today, but when that bacteria mutates to survive, which it always has and it always will, that antibiotic no longer works. So rather than be reliant on natural processes, we designed ours with first principles in mind. Former head of research of Johnson & Johnson Australasia synthesized a compound that has multiple mechanisms of action, that's attracted and binds to any bacteria, gram-positive, gram-negative, or their hyper-mutated form, and most importantly, keeps on working with repeated use. Anybody can have a new antibiotic today. You've got to have one that works and keeps on working, the fundamental challenge of antibiotics as we know it. So our lead candidate is focused on sepsis, septicemia, or blood poisoning. It is the #1 most expensive condition treated in health, double the second most expensive. There's around a 50 million patient population each year that become infected. And the statistics as we know it is 1 in 5 who develop sepsis, and I've developed sepsis, thankfully I survived, but 1 in 5 do not. And in fact in the hospital environment, 1 of 3. And therefore it is the most expensive condition to treat, and there are currently no drug therapies specifically for sepsis. The reason why that is drugs or antibiotics are designed to attack one type of bacteria. Now, when a patient presents, as it was the case with myself, the clinician quickly does a pathology blood draw, they rush it off to the laboratory, and they cultivate out that blood to work out what type of bacteria and what type of antibiotic it's susceptible to. Well, that is archaic guesswork up front. They administer a cocktail of antibiotics in the hope that it's this antibiotic for that type of bacteria. Well, because every hour left untreated, the likelihood of survival decreases by 6%, and that cocktail is absolute guesswork. We designed ours to attach, bind, and interact with any bacteria in the blood. We don't care whether it's a gram-positive, say a staph, or a gram-negative, say an E. coli, but most importantly, we don't care, or the compound doesn't care, whether it's in a hypermutated state. And that really allows that time-consuming clinical guesswork up front to be a thing of the past, because the faster you're on to that patient infection, the better the patient outcome. Here we talk about a hypothesized mechanism of action. Now, it's a pretty picture in this instance. It's based upon hundreds of pages of research report, which was, I think today, actually published in an abstract. But in essence, when we look to the bacteria, in this case E. coli, it's hypothesized that it binds to the outer cell of the bacteria itself. It enters the bacteria, so rather than puncturing or dissolving, there's nothing harmful about it, it actually enters the bacteria and shuts down the energetics of the bacteria, which is the ATP production, or effectively heart. What happens when that occurs is it does two things. One, the cell is unable to continue to divide and cause harm. And two, the cell is not able to come back to life and go through the normal bacterial processes. As we see in stage 4 there, and we see in our next page, in the higher concentrations, we're taking advantage of the high metabolic processes that occur within bacteria. They're the only cells in the body that have around 10 atmospheres of pressure. So that level of specificity between a healthy cell, which has no internal pressure, versus a bacterial cell is some of the mechanisms of attack or interaction that our compound uniquely has. This is our phase 1 intravascular study. In essence, it's single dose escalation, intravascular escalation into the veins of humans, approximately 80 humans. They're healthy, you or me. There's nothing interesting particularly about them, except we do dose escalate amongst those humans, really with the goal of seeing how much compound can that human population take. And should have mentioned there, of course, that interim data should be available approximately Q4 of this year. Our second clinical program, which is really focused upon efficacy, yes, it's a collapse phase 1, phase 2, but looking to efficacy, is a clinical program sponsored by the West Australian Health Department. We actually don't pay for it, it's fantastic, caveat free. The trial investigators there are some of the world-leading experts. Professor Fiona Wood, former Australian of the Year for her burns work. There we have two essentially cohorts of persons. The bacterial types that these patients have are sometimes multi-drug resistance, sometimes they've suffered these bacteria for many years, and the clinical insights that we will gain, it's happening as we speak, but the interim data is expected to be available in Q3, could really be a pivotal moment where we start to see that challenge of antibiotic resistance or multi-drug resistant infections to potentially be a thing of the past when extrapolated out to large patient populations. And here, of course, we talk about the size of those populations. So utilising the compound, now why are we able to work in the blood, topically, or to hear the opposite acidity of the study, the stomach, the full pH range? And that is because our compound is water soluble across all pHs. So Helicobacter pylori, keeping it really simply, is the bad bacteria that exists in the upper duodenum. The good bacteria exists in the lower duodenum. It is a major patient population, and we only focus upon unmet medical needs, of which Helicobacter is currently serviced by what's called a triplicate formula, where you basically take 3 aggressive antibiotics and you're really, keeping it simply, kind of blow out your gastrointestinal tract and get rid of the hazardous bacteria. We're seeking to do that via one formula, being our own, and if we look to the studies of preclinical data to date, I think this is in rats and from memory at a local institute, there we compare against amoxicillin combination. We basically see that we reduce the bacterial colonisation in the animals quite dramatically. And as we see on the next page, we don't seem to harm the healthy good bacteria. So why is that? Well, our theory is essentially that because the bad bacteria in the upper duodenum, we're interacting with them up there, and when it gets through the metres and metres down to the good bacteria, that water solubility appears to have either broken it down as a compound through the bacteria or broken it down as a water-soluble solution. So we are halfway through those preclinical studies, which is led by a world-leading H. pylori expert, Professor Phil Sutton, and those studies are happening at Murdoch Children's Research Institute out of in Melbourne itself. I believe we will have preclinical data over the next month, which, if successful, would be pivotal to continue to advance this compound into the human stage. So this is starting to look as our compound as a technology. We talked about it from an antibacterial perspective. Now we start to look at it from an antiviral perspective. Don't use antibiotics for viruses. It's not advised. We call this an antibiotic simply for means of communication. It's really an anti-infective. Here we talk about it against COVID. We were selected as a priority 1 test candidate by a leading government research institute in Australia, and in fact the Australian government itself. This study is assessing 2 compounds. One is our traditional antibacterial compound, Recce 327. The other is Recce 529. That is because we synthesised the process to have a higher affinity for the proteins of the envelope cells of the virus itself. And in this organoid model, which is basically where you get healthy human tissue, you infect the human tissue with COVID, we demonstrated significant dose-dependent reductions in the COVID virus without harming the healthy human tissue. This is looking to our compound in an Australian study, by example. Here we demonstrated significant 99.9, well, we've actually done 99.99% reduction in the COVID virus, as we see on the graph there. But most importantly, anything can kill COVID. You don't want to kill those healthy cells, and that's where you see cell viability remaining effectively normal. Very, very important, meaning it was a specific interaction, which is what we absolutely seek. This study was an overseas study done in the United States. This is starting to look at the compound from effectively a 5th method of administration. So a nasal administration to get into the upper respiratory tract of hamsters, which is a gold standard model when it comes to coronavirus. There we see moving from a high colonisation of the virus itself in the respiratory of these animals through to a very low colonisation. And in fact, I think when we look at a 3-log reduction, we're looking at a 99.9% reduction in that virus. And there's studies continuing in the background related to resistant forms of that COVID virus. We maintain a privileged position in our intellectual property in that this discovery is wholly owned. It's not in-licensed, it's not teetering on the edge of somebody else's balance sheet. This is our technology as created by a former head of research of Johnson & Johnson Australasia of a decade, and he was actually executive director of their board. So this land grab opportunity is a composition of matter patents supported. Our patent family 1 is manufacturing and curative claims. Our patent family 2 is preventative claims, as well as various methods of administration: intravascular, oral, topical, nasal, inhaler, protected in all the major markets of the world. And patent family 3 is our viral patents, so coronavirus, Ross River virus, influenza virus. And as we see in the transition from allowed, I would anticipate them naturally becoming granted very soon. This is actually out of our Sydney Macquarie Park facility. Here we manufacture our own product. We actually synthesize it through a very, simple method of manufacture. We have a very economical method. We have no expensive raw materials. We have no expensive waste. We're producing to GMP specification and volume in approximately 500 doses per 1 hour of production. And we're all set up to continue to support that through these phase 1, phase 2, and potentially phase 3 trials thereafter. As a company, we are obviously Australian Stock Exchange listed. If anyone prefers trading in euros, we're also available on the Frankfurt Exchange. Our market capitalisation is around $180-ish million. We have around 23 million, or we had around 23 million cash at bank at the end of March. For us who saw that quarterly, that represented around 33 quarters of cash. That's extraordinary. That's like 8 years of money. I assure you we're not going to sit on that for 8 years. Our goal is to bring on additional clinical programs. So needless to say, be assured we're not doing a capital raise tomorrow. There we have, in the top 20 shareholders, about a 56% holding or founder holding. Our institutional holders are groups like Fidelity International and others. We have very few options on issue, very well funded, and obviously an excellent intellectual property portfolio. Just last week, a leading healthcare equities research group came out with a research report. Everybody likes to spruik such a thing because it's always very favourable as to valuations. But what I liked about this one is it really captured in a simplistic form the value proposition of a key infectious disease asset, a major unmet medical need, and a solid cash balance sheet to support its clinical development. And I'd encourage you to look at that in the background. I think it's on our website. So really, in summary, as an infectious disease business in the midst of a global infectious disease crisis, we have a strong portfolio of infectious disease candidates. We have an evolving appreciation of the importance of new medicine. We have a strong balance sheet to continue to support our development. We have 2 clinical candidates which are imminently to provide news flow to support our what we would believe our continued development down those clinical pathways, and a robust financial position to support. I'm James Graham. Thank you very much for listening, and welcome speaking further in the background.